professional arena participate as equals in a nation-building project. When we talk about education, it is not merely information stored in the brains of a student. It stays there unused, unutilized for the rest of their life. There has to be life-making, character-making assimilation of ideas. We are privileged today and immensely delighted to have Professor Christine with us. She is working at Medical University of Graz, Austria, as a professor of interactive microbiome research. She has done her PhD at the University of Regensburg, Germany, followed by 2 postdoc positions at the University Hospital in Regensburg, NASA Jet Propulsion Laboratory, USA. Her expertise is in the area of human and hospital indoor microbiome with focus on archaea bacteria. Her research work is well appreciated and published in journals of high repute. She is a wonderful speaker who has also delivered an insightful TEDx talk on why we need to worry when we send humans to Mars. Today, she would be talking on microbes in space, a microbiological perspective on planetary protection, human spaceflight, and extraterrestrial life. I now invite our distinguished speaker to share her profound knowledge on the matter with the audience. Dr. Echinger, over to you. Thank you.
Thank you very much, Dr. Kumar, for these very nice and kind words. Thank you, Kriti, also for inviting me. I feel very, very honored to be amongst you and to open this colloquium with my talk. I will try to share my screen. I hope this works, and then begin with my talk. I hope you can see it now.
Yes, it is visible.
Okay, so very good. So I'm very excited to be able to talk about one of my most favorite topics, which is microbes in space. And I hope I can convince you that it's very important to think about all the topics related to microbiology in space, not only when we send humans, but also maybe about planetary protection and all the other things that are coming along. So I hope I can fascinate you also to think about this topic even more. So what are my topics today? Of course, the most intriguing question human mankind actually has is whether life exists beyond Earth. That's why we are doing all these missions. We were sending spacecraft, for instance, to Mars. We try to understand whether there is life out there. So whether the human or the Earth life as we know it is the only life out there, which is maybe not really true. So, but before we do so, and before we think about life out there and we try to find it, we need to learn more about life that we have actually on Earth to maybe get an idea how life outside could actually look like. So we need to analyze our own habitats, our own biotopes of Earth even more. Then the second topic I will be talking about is planetary protection. So we need to think about whether contamination deriving from our Earth could harm life or biotopes on other planets. And then at the end, I will be talking about human missions and their microbiomes and why human missions are even more complicated than robotic missions. So, but let me start. So if we try to find life, let's say on Mars, we have to have something in mind. So you cannot just go there and look for some biomolecules, random biomolecules, for instance. You have to have an idea what you're looking for, because otherwise the search might not really work. So what you can do, even if we cannot access other planets really easily, we can try to understand the life of Earth in more thorough way. So in particular, you can look at sites which are similar to extraterrestrial sites. For instance, so-called analog sites. Analog sites are sites that we find on Earth, which have similar characteristics to extraterrestrial biotopes. For instance, they're very cold, they're frozen, like permafrost environment, or you have very high or low pH, or there's a lot of salt. So in this project, I'm just showing on the screen, which is called MACE, Mars Analogs for Space Exploration, we specifically looked at those extreme environments. And we particularly looked at extreme environments which do not have specific oxygen, because most of the extraterrestrial environments, as we know them, do not have a lot of oxygen available. So we specifically looked at sites which have low oxygen availability on Earth, and which have implications for space life, maybe if it's out there. So we looked, for instance, at sulfuric springs, which resemble early Mars. That was our opinion. We specifically looked at hypersaline environment, which is here the Bolby mine. It's a mine underneath the surface of the United Kingdom, and you can find a lot of salt-loving microorganisms there. We also looked at acidic lakes and rivers. We looked at glaciers and permafrost environments. So, of course, as you can imagine, all of these environments have a completely different microbiome, right? They are having a huge load of microbial diversity. And we were particularly interested whether there is a core microbiome. So if there are certain microbes that are overlapping, and also with respect to the functions. So which functions do these microorganisms have to adapt to these environments? Overall, we did more than 1,000 different cultivation assays to also get some model organisms for the laboratory to be able to do some physiological tests. And we were able to isolate 31 model organisms out from these different sites. We also derived additionally 15 high-quality genomes, which are currently analyzed. So without going further into detail into this diversity I'm showing here, I'm showing you the big outcomes of the story. So on the one hand, we found that basically the acidic lakes and the hypersaline environments were very similar in their microbiome composition. This was something which was very intriguing and interesting for us, which is very insightful also for some Mars environments maybe, where we find both of these conditions combined. So obviously there's a certain type of microbiome similar to both sites and having certain overlap. This is completely interesting to us. On the other hand, we also looked at the specific functions, as I mentioned, and we saw that indeed these environments that we selected for our study are extreme for microorganisms. So it's difficult for these microorganisms to survive in these environments. In particular, we found increased resistances with respect to certain metals. They were higher in cold shock resistance. They also showed higher increase of multi-drug resistance efflux pumps, for instance. They also dealt better with periplasmic stress, and so on and so on. So we found a lot of implications that indeed the microorganisms have to specifically adapt to these extreme environments. The most intriguing part, however, was that we found that these microorganisms in these extreme environments were specifically adapted to organic use, which means that they had less capability to do CO2 fixation. This was really amazing because we actually thought we are focusing on Mars-like environments. We're excluding the oxygen, which gives us much more information about habitable environments on Mars. But obviously, we found that these microorganisms are very much adapted to the high organic load that our Earth atmosphere actually provides. And this makes us thinking about whether it really makes sense to use extraterrestrial on Earth, those analog sites, to get information about extraterrestrial life. So we always have this problem. Earth is not like an extraterrestrial planet. It is full of organics, in most cases also full of oxygen. So we only get a glimpse of an idea of how life might look like on another planet. So this was actually just a little bit of a journey into this project I wanted to show you, but I'm now going to talk a bit more on other topics. The next topic is planetary protection. So the question here is whether we have to protect the extraterrestrial ecosystem and to make sure that the life detection mission has success by not contaminating it with Earth life. So what is the background? So as you all know, and you're very aware about these recent success of life detection missions that are going to Mars and the new ideas that are coming up for the next years, we are really super interested in finding life out there at some point, right? So there's not only Mars, which has liquid water, as we know since a few years. There's also Jupiter with the moons Europa and Ganymede. There's also Saturn with the moon Enceladus, and so on. There's so much places where we could potentially expect life. And this is super exciting, and we really would like to learn more about it. And that's why we're sending missions, right? There's the idea to send, of course, robotic missions, but at some point we want to also send humans. So, but if you want to look for life on another planet, and this is the goal of the mission, you should not bring your own life with you from Earth, because you would cover all the signatures from the external life or the extraterrestrial life, which makes your mission like not being successful. So planetary protection is the background or the study type of understanding how much contamination we are actually bringing to other planets and whether this would harm our mission success. On the one hand, you have forward contamination, which means that you're taking microbial life or also biosignatures of them to another planet. But there's also the concern of bringing maybe contamination from other planets back to Earth. So there's still a possibility that these planets have certain life, and it might be a problem if you bring such life back to Earth, right? So you have to consider both directions. So what you can do, of course, before you send a spacecraft, you're just making sure that there is no microbes on it. It's not so easy, actually, because the missions that we are building at the moment, they are highly complex technical instruments, and you cannot just bake them. You cannot put them into an oven and really heat them up. This was actually done still in the 70s in the Viking missions. So those were baked, those were completely heated and sterilized before sent to Mars. Now the missions are different. They are very sensitive. You cannot bake them. So you have to think about other strategies to make them microbe-free. So what you do is basically you construct those instruments and test and launch them under complete biocontamination control. So you build those instruments in clean rooms and you make sure that they are not getting contaminated at any time point. So, but it all depends on the goal, on the goal of the mission. So where you want to go, because it makes it different if you're going, for instance, to some asteroid or some moon, which certainly does not harbor any life. But if you send missions, for instance, to Mars or any of the moons that I just mentioned, you have to make sure that your instrument is not contaminated to a certain level. So there's different categories. For instance, usually if you have a flyby or an orbiter for Mars, you're in category 3. If you're trying to land, it's already category 4, which means that there's a lot of more rules that apply. By the rules for planetary protection, it's actually all fixed in a certain treaty. It's called this Outer Space Treaty, and all nations that are actually going to outer space signed this treaty at some point. So it's mandatory before you send a mission to somewhere else to stick to the rules of this treaty. And this also includes the certain amount of spores and microbes that can be or are allowed to be transported by such instrumentation. So this is the problem that we are actually facing. So we are all living on a microbial planet, and the microbial planet is full of life. So there's a lot of microbes all over the place. So it's not so easy to keep a spacecraft really, really clean. One other problem is that we are actually, the humans are spreading microbes all the time. It's thought that we are spreading about 37 millions of microbes per hour. So we are all having a certain microbial cloud around us. This is not a problem in general, but it's a problem if you try to build a sterile spacecraft. So if you're close to the spacecraft, and all the engineers that are actually so close to the spacecraft have to be protected not to spread their cloud of microbes onto the instruments. So how is this all done? It's all important to have certain contamination control, for instance, in clean rooms. So for many space missions, there's certain clean rooms that are built to make sure that the microbes are kept out or not close to the instrument. So the clean rooms, which means also that the people working in those clean rooms have to be in certain suits, they have to be trained how to move in such a clean room, not to touch the surfaces and so on. And on the other hand, there's also control. We are taking thousands of samples of these instruments, of these spacecraft, to make sure that the contamination limits are not exceeded. So these clean rooms, which are used to build the spacecraft, are kind of extreme environments. So they are difficult to survive for microbes anyhow. They are completely confined. There's airlocks all over, so you cannot just easily enter or put stuff inside. And there's a strict control of particulates and also, of course, the biocontamination. As I mentioned already, the team that is working in such clean rooms have certain regulations. They have to put certain clothing, and they do a lot of cleaning inside to make sure that the contamination is not rising. The interesting thing, is that these environments are very selective for survival specialists. So certain microbes that are adapted to these environments, they don't care. For them, it's still easy to survive in such environments, and those microbes also bear most resistances. Though they are resistant against the cleaning agent, they are resistant against desiccation and so on. And so they are carrying a lot of risk also to be able to survive on another planet. And this is something we have to control. So this is an example of a clean room in Turin in Italy, which was used for, in our case, the ExoMars 2016, also used for the ExoMars 2020 mission. And you see that there is basically a changing room in the middle. So people come already with changed clothes there. They change clothes again. So they're going through a two-step clothing procedure. And then they are able to enter the clean rooms, which are actually harboring the spacecraft. As I mentioned, the teams undergo trainings and medical checks to make sure that they are not spreading too many microbes on the surfaces of the spacecraft. Also here in this clean room, there's also microbiology laboratory on site. So people do a lot of checkups of the spacecraft during the procedure of building it. Just showing a few examples. For instance, what you see here is basically some samplings that we have done before the spacecraft was brought into the clean room, and after or when the spacecraft was actually inside. And you see 03 is actually the garment area, so the clothing area of the people. And you see before the spacecraft it was there, a lot of transfer from there was observed into the clean room area. So a lot of microbes were brought into the clean room area. We recognized that and performed additional trainings and cleanings. And after a while, you saw that basically all the microbes, or most of the microbes from the clothing area, were really kept in the clothing area and not transported into the clean rooms themselves. So this was a full success. So when you follow a spacecraft, you obviously also hope that the spacecraft will arrive at the planet at some point, the target of your mission, basically. I'm just showing a sad picture of the Mars 2016 mission that actually dropped on the surface of Mars, so it did not reach the surface in complete manner, which was for us a real pity because we did a lot of analysis of the spacecraft before, but at least we were sure that we were not contaminating the surface beyond the allowed borders. But as you know, a lot of missions now, 2021, actually arrived at Martian surface as well. And all of these also have been under these planetary protection restrictions, and we are not fearing a lot of contamination coming from them, and yeah, avoiding the success of the mission. So I think that was everything I wanted to tell you about planetary protection. So I think you get a little bit of a glimpse how complicated it is, also seeing from a microbiology side to send spacecraft to another planet. It's not so easy. And also, actually, it costs a lot. So it also has to be implemented in all the budget plans and everything, that the planetary protection is done in a correct way. So, but now I would like to switch to the question of how can we send humans to Mars and other places, and how do we deal with the microbes they are carrying and the environmental microbes that are also in the spacecraft. So how do we deal with human spaceflight and their microbes? Why do we have to think about that in any case? Well, on the one hand, of course, there's a possible risk that microbes bring. Of course, it's obvious that they could attack the health of the crew members, right? As we all know that there's also pathogens, microbes, of course, can make us sick under certain circumstances. But on the other hand, we also know that microbes can degrade surface materials, so they can attack the integrity of the spacecraft. This was found actually many times already, for instance, for the Russian space station Mir, where it was found that microbes actually attacked the windows already. So they attacked the outer rim of the windows, making it not very tight anymore. And also they attacked certain wires, and there was problems with the electricity at some point, which actually led to the point that the space station was given up and left in space. So microbes can do bad stuff, and this is something which is not good when we are sending people out to Mars, for instance, and back, which would take more than 500 days for sure. So we have to control those microbes somehow. But also the human itself is a problem, because the human immune system is compromised under space conditions. This is frequently observed under astronauts. They really have a reduced immune response due to the stressful situation they are going through. On the other hand, we also know that microorganisms can be affected by microgravity per se. Some of them react with higher virulence, which is not good, because it means they make us maybe sicker. And even if we have already a reduced immune system, this is not a good situation, right? And we also have to think about the efficacy of antibiotics and other medications that are used in space. Some of them show a different effect on the human, and the medication might even be decreased in efficacy under these conditions. So there's a lot of things that we have to study when we think about infections and all that part. So it is quite sure that we cannot go to space without microbes. We have them with us, so we cannot kind of put the astronaut apart from its microbiome. It's not possible. Also, we need microbes for the regeneration modules aboard, so for the recycling of water and so on. Also, the plants that we are bringing to space basically also need their microbiome, so we cannot leave microbes at home. That's not working. So we need to find the balance of control and the countermeasures that we are trying to do. So controlling and understanding, monitoring is necessary. But of course, we cannot wipe out all microbes. So for that, we need a lot of research. So when I'm talking about research with respect to microbes in space, you completely understand that actually the settings that we're looking at are very restricted. So we have the space station, we have certain small human missions that we can actually analyze. Also, the number of astronauts, cosmonauts, spacefaring people is really, really low. So we're restricted in understanding or getting the real pictures of microbiology in human spaceflight. So what we are doing is we look specifically also at simulations of spaceflight. It's never the same for sure, but at least we get a glimpse on what's ongoing with respect to the humans and their microbes in closed systems. One of these closed systems we have looked at is the HI-SEAS 4 mission, which is an isolation study. It was initiated by NASA and also supported by NASA. And actually, it is an isolation study which took 1 hour, 1 year. Sorry, 1 hour would be fine. Actually, 1 year is really bad. So there were 6 people completely enclosed in this environment. It's called a dome. I will show you a video in a few seconds. And it was very close, very tight for these people. And they spent 1 year enclosed in this dome. They were not allowed to have any contact to any other people. They were allowed to get out of the dome, but only wearing a spacesuit. And they were basically completely simulating a stay on the Martian surface. This was actually taking place on Hawaii, I have to say. So the background is also like Martian similar. So I brought a little video and I will show you a few parts of it to get you a bit an idea of how it looks like inside. I have to actually give credit to the HI-SEAS crew, which provided this video for us. So you see here a bit of the inside. So this is the joint living area. Upside, you see basically all the living areas and the bedrooms of the crew members. So you see also how small it is inside. There's not much room, not much space. This is the spacesuit, actually, by the way. You see some working desks for the people here. You see also a lot of working out areas like the treadmill. So they have to stay fit, those people. And I'm moving a bit forward. You see also a bit the kitchen here. And actually there's also a laboratory attached for the analysis they are doing inside. So we were very interested in being part of this mission and to get samples, microbiology samples from inside. And actually we got samples from the different surfaces, so from kitchen desks, for instance, from the floor, but also from bedroom areas. And we also got samples from the humans, from the people that are working and living there. And we were very interested in understanding the dynamics of the microbiome. So how is the exchange of the different microbiomes? Can we see a change over time as well? And we analyzed all those samples. As I mentioned, skin and environmental samples. So what we see here, basically, it's a measure of the diversity of the microbiomes on the surfaces, which is always shown in blue. And the orange ones are the ones from the crew. And what we see is that the diversity of the microbiomes on the surfaces remained really, really stable over time. But the microbiome diversity of the crew changed over time. This was kind of something completely unexpected, which we did not understand very well at the beginning, because you would expect that actually the surface's microbiome is much more dynamic than the human skin. But obviously the human skin was able to get microbes from the environment and accept it in their own microbiomes. So we also saw a lot of difference with respect to the development over time. So actually there were different phases of development on the microbiome level. We saw a lot of homogenization, so the samples became even more similar to each other. But we also saw fluctuation of certain microbes. For instance, the Acinetobacter, which rose a lot in the last month of the entire situation of the mission. Staphylococcus was changing all the time, which was highly fluctuating. And there was also Methanobrevibacter and Archaeon that we actually carry in our gut that we mainly observed at the very beginning of the mission, obviously. And we also found out later there were some sanitary problems with the toilet, and obviously there were some spreading of microbes from the toilet into the area, which is not so nice to think about, right? But we could see it also in the microbiome patterns all over. What we could also see is actually that certain crew members are having more contact to each other than others, and you can see these in these microbiome profiles. So basically, each of the crew members has a certain color, and you see, for instance, that crew member A was interacting a lot with crew member F in the first month, but not anymore afterwards, and that there's certain people also spreading more microbes to other people than certain others. So this is kind of interesting also to understand the team dynamics inside, and that we can also see this on the microbiome of the skin, that some people have more contact than others. We also analyzed certain phenotypes of the microbiome, so we were very interested in whether the microbiomes inside form a lot of biofilms or whether this is changing. Basically, we saw a lot of fluctuations here. We also looked at pathogens that are kind of, if they are rising over time, they did not. It was also a fluctuation over time, and so on and so on. So all over, it's very dynamic. It needs basically a lot of monitoring. Also, when you think about sending humans to Mars at some point, because this has to be under control just to make sure that there is not an outbreak coming from somewhere. So as I mentioned, what did we learn? We learned, and this was amazing for us to see, that surface microbiomes are largely stable, but that the human skin microbiome really fluctuates and is able to accept microorganisms from the environment, but also from the crew in large amount. And this, of course, has some impact on future human spaceflight and about the conceptualization and also the monitoring that we have to plan in the future. So, and the last topic I'm trying to discuss here is the International Space Station, and one of the studies that we did there, which is the best model for spaceflight as we have right now. So we have not sent humans much beyond the space station yet, and it's very important to study the space station as thorough as we can. So I'm starting with a cartoon which was published a few years ago. It says, "Dear bacteria, I'm happy to announce that we are harboring on the International Space Station too. And the most important part is we reach there without any training or facility." So yeah, because the microbes go with us, with the humans, with the cargo, which everything that we are sending to the International Space Station is bringing also microbes, and these microbes have to be studied and have to be understood to make some implications for future. Why is the International Space Station so interesting for us microbiologists? Because it's the most confined environment that we actually can look at. So there is no windows that you can basically open, let certain microbes in from plants, from your environment. There's only a bit of an exchange of cargo, so there's only a little bit of a supply coming from time to time. And there's, of course, the crew, which is exchanged 1 or 2 people every 3 to 6 months. But the rest is completely stable. And this is really, really interesting. Also with respect to the similarity to other ground-based environments that we are seeing. For instance, if we think about the managing of intensive care units at the clinics, which is also kind of a restricted environment where we also try to control the microbes inside. When we think about operating rooms, so everything should be closed and no microbes should come in from the outside. Same is true for clean rooms, where we try to protect, of course, the substances or the products inside. So all of this has something in a similar manner, and we're trying to find out how to control these environments, and to make it a healthy environment also for the humans. So I'm here particularly talking about one mission I also already mentioned. It is called Extremophiles. This is a project that we actually performed on the International Space Station. It was proposed and accepted in 2009, so a really long time ago, and it flew in 2017, which actually tells you how long it can take to implement a space station experiment. So this is something you always have to keep in mind. We have a lot of ideas and we would like to do a lot of studies in space, but it takes a lot of time, it costs a lot of money, and it's very complicated to do. So what did we do? We had basically 3 sampling sessions, A, B, C, and the astronaut, in this case it was Jack Fisher, a US-American astronaut, took samples for us, and you see him here wiping different surfaces with the wipes that we actually provided. And I also brought a little bit of a movie to show you how this works in the International Space Station if you would like to sample the environment. So I hope you can see him. This is actually Jack Fisher preparing for the sampling. Oh no, it's working. Okay. So it's, as you see, complicated. He has to read our manual that we provided, and he tries to get an idea what he needs to do and where the stuff is. And he has to put everything under certain clamps to make sure not everything is flying away. And so he's now still preparing and moving a bit forward. And at some point you will see that he's starting doing sampling. He's now gathering the gloves from the bags that we provided. It's all very complicated. It takes a lot of time, still doing the gloves here. And now he's putting on the glove. And then he's moving. All that is also complicated in space. So he also has to make sure that everything is ready. This is the wipe that we provided. He's trying to unfold it, and then he is wiping the surface of this return sensor grid housing, where actually all the dust is sucked in and the air is filtered. So I'm moving a bit forward now. Even if it would be great to follow him for hours, I guess. I think it's very nice to see him floating around. But the samples that we have been taken actually from different surfaces, from light covers, from laptops, from hand grips, from the sleeping units. We also got samples. We got samples from the treadmill, from waste and hygiene compartment, and dining table, all of that. All these samples came back into the laboratory, and you see part of my crew members here on ground who analyzed those data from those samples. So without telling you too much details, I just put some very important findings to the table. What we found is that the ISS microbiome, similar to the high seas microbiome I showed you before, is also subject to fluctuations. Even if you have a core microbiome, so certain microbes that stay there for tens of years, as we know now. So for instance, Bacillus stays very stably in the International Space Station. But there's fluctuations because of the different cleaning regime, because of the crew and how the crew behaves and all the other things. So it's not stable, there's fluctuations of the microbiome. And all of the different locations in the International Space Station, they have also their specific microbiome. So it is important to understand, of course, that the waste and hygiene compartment has a different microbiome compared to, for instance, the laptop that we analyzed or other areas that we looked at. The most important and the most abundant microorganisms that we find are, of course, human-associated microbes, for instance, Streptococcus, as it's found here, but also Corynebacterium. And I mentioned that we also very frequently find Bacillus, in particular from spores that we can cultivate in the laboratory. So as I mentioned already, there is a difference of the locations within the International Space Station, and we can also see that in particular the personal, so for instance the sleeping unit, the personal areas really have the highest diversity of microorganisms that can be found. So the more there is an influence of the humans, you find a higher amount of microorganisms there. And in this part of the study, we actually compared the International Space Station microbiome to ground environments. So we particularly used indoor environments, but also human microbiomes to compare the composition of the microbiome. And we found that basically the International Space Station is not much different to a ground-based environment. In particular, it resembles a bit the doorknobs of a bathroom or bathroom microbiomes in general, because there's a little bit of moisture in the International Space Station. Of course, most of the microbes that we are finding are from the humans. That's similar as it is the case in your own bathroom. So we also looked at the resistances of these microbes, because there have been some publications mentioning that there is a risk that these microbes on the International Space Station have higher amount of antibiotic resistances. They might be more harmful to the human. And this is something we found very interesting. And we actually compared also that. And we found actually that the microbes on the International Space Station, despite the stress and all the circumstances, are not more resistant. They are not higher in virulence. They are not posing per se a higher risk to the crew than those microbes do, which are surrounding us on Earth. So there's not a big difference to the Earth-like microbiomes, and not a clear risk for the crew as we could identify that. So this is also shown as we looked at the antibiotic resistance profiles of isolates that we retrieved from the International Space Station. Also here, these profiles were very similar to the profiles that we see from close relatives from ground isolates. So no reason for concern for the crew members at this time point. But what we also saw is that these microbes are very interested in interacting with surfaces. So they can attach to surfaces, they like to be on surfaces, they can resist metal surfaces also very well. So it's actually a little bit of a problem, as we observed in the International Space Station mirror. So they might be attacking the surfaces somehow. And this is what we see here, is basically that the ISS microbes really attach to surfaces. And in this case, it's Nomex. Nomex is a textile that is used for the big bags that are used on the International Space Station. And the microbes here, as you can see, they like to attach to these fibers. So this has to be analyzed even further. At the moment, there is no direct concern with respect to the crew health, but biofilm formation and material integrity might indeed be a concern with respect to the microbes, which we have to study in future. So I'm coming a bit already to the conclusion and the outlook, and I'm trying to share a few ideas with you about what we are thinking we have to think about in future. On the one hand, of course, we need to think about how we monitor human health during space flight and how this impacts also the forward and backward contamination, as I mentioned, in particular when we try to send humans forward and back from Mars, how this might affect all the planetary protection considerations. We need to think about what is a healthy microbiome. So what do we need to know about the astronauts before we send them, about the microbiome, about the stability of the microbiome? And how can we implement even further that simulation studies that we do on ground to learn more about the microbes inside and the humans which carry those microbes? We also have to think about the methodology. So if we want to screen the microbiome, the microbiology in the spaceflight settings, how do we do that? So you cannot easily do a DNA extraction. You cannot just do a sequencing so easily. But maybe the MinION strategy is something that we can follow. Nanopore is actually already used on the International Space Station, so it seems to work, and this might be applicable also in future. Yeah. There's also the question of the risks, of course, for the human, whether the microbiome, the gut microbiome, for instance, reacts with perturbations and dysbiosis, which has to be also treated, and there has to be a therapy at some point. And how you do this therapy, all these things are very complicated to think about. The schedules, yeah, how if you need a quarantine for the astronauts, how we do that, how do you do it with astronauts coming back from the space flight, and so on. There's a lot of things that we have to think about. This is, I think, the point where I would like to end my talk. I hope I could give you a lot of insights in how complicated space microbiology is to do, but there is a lot of research as well needed, so maybe I could interest you also to jump into this very fascinating topic. And I thank you for your attention. If you like to follow me on Twitter, there is my Twitter handle as well, so maybe there's more interested people joining me. And now I'm open for all of your questions. Thank you very much.
Thank you, Dr. Christine. Really a wonderful presentation. And now we would be opening our house for question answer session. For this, I would invite Dr. Manpreet Kaur Rawal to please moderate the session. Manpreet?
Yeah, thank you, Dr. Kriti. Well, Dr. Christine, you were really awesome in your this presentation, and it was really a complicated one. I must say that this spacecraft and space microbiology is really difficult to handle. So we have a few questions, and here we have Dr. Sunil Srivastava, and he wants to ask a few questions from you. So I'll allow I'll ask Mr. Anuj, allow Dr. Srivastava to unmute so that he can ask question by himself. So Mr. Anuj?
Can I ask? I think I'm on mute.
Yeah.
Okay.
Yes, sir.
Okay. Herzlich willkommen in Indien, Professor Christine.
Thank you.
Danke sehr für den sehr guten Vortrag. Ich habe eine Frage. Bitte. No? So, I was just wondering, can I speak English now?
Yeah, please.
Because my Deutsch is not that good.
No, your German is perfect, but I think you're losing the others, right?
Yes.
Okay, thanks. I was just wondering, has the Martinian soil have been examined for some microbes? Because the Martinian soil, they have found water, I believe, and it is exposed to a lot of radiations. So do you find anything like Deinococcus radiodurans or something like that?
That's a very fantastic question. So, of course, on Mars, it has been tried to analyze whether there is life already, but at this time point, we do not know. So all the information on the microbiology we could survive in the Martian soil actually stems from simulation on ground. So we know quite well about the composition of the Martian soil, and we can produce that soil on ground. And there were a lot of exposure experiments with certain microbes. And we understood that even if the radiation is very high on the surface of Mars, only 2 centimeters of soil is enough to shield completely from the radiation. So if you kind of have microbiology deeper than 2 centimeters, it's very likely to survive there. Of course, the soil is very bad to microbes as well, and not all will survive, but there's, as you mentioned, also liquid water at some places, and this is what actually the rovers will look at and analyze in more detail if we find those biomarkers for life.
My another question is, have you found any nucleic acid as such in the soil, some debris or something? Like we have?
No, not yet.
Not yet. Okay, thank you.
That's also a very interesting point, actually, that the human mankind has not tried to find life on Mars since the Viking missions in 1970s. So all the other missions that we have sent to Mars to this point until now actually tried to understand the geology and other things on the Martian surface, and they did not look for life. It's very interesting.
And in Europa also we have, what is it? In the Europa satellite of Jupiter?
Yeah.
We have lots of water, I believe.
Yeah.
Okay, thank you so much. Vielen Dank.
Thank you.
Thank you, Dr. Srivastava. So we have a few questions from chat box. So let me take one by one. So first is, were microbes also present on outer surface of ISS? If so, weren't they harmed by the radiations of sun?
Yeah, that's a good question. So it's about the outer surface of the International Space Station. So there is not a single sampling except one Russian sampling that has taken place outside of the International Space Station, which was super complicated to do because there is, well, it's super cold outside. So if there is microbes, they will certainly not be able to grow there, right? So there were samples taken from the outside, and they found indeed a few DNA molecules from microbes that were very similar to the microbes from the environment in the US American facility where the ISS modules were built. So obviously there was some microbes still attaching to the outer surface of the International Space Station from the surrounding in, I think it was Florida or any other place close to the sea in the US. But there was no other signatures since then that really make sense that have been found about extraterrestrial microbes or so. And it's for sure only it's the nucleic acid, the microbes will not be able to survive under full radiation outside on the ISS.
Okay. So we have another question. How astronauts suppress microbes on ISS? Is there any different method used by them?
So basically the astronauts, if they try to clean or to control the microbiology on the surfaces, they do it as we do it. So they have one cleaning day per week, which is usually Saturday. So similar, I think, to most people anyways. Once a week there's cleaning day, and they do vacuum cleaning, and they do surface cleaning, and they also do disinfection when it is necessary. So it's not really, how can I say, very clearly explained when they should clean very thoroughly. It means that everything should be kind of visibly clean. The problem is, as you saw maybe in the video, I've shown that the spacecraft inside is really difficult to clean because you have a lot of cables, you have laptops, you have a lot of different surfaces. So there's no floor which you can actually clean very easily. So you cannot wipe areas very easily. So it's very hard to keep it clean. But yes, they do it similar as we do on ground. At least they try.
Okay. So I read that there is another question. I read that Martian life underwent extinction twice. So if we didn't find any nucleic acid on Mars, how did they come to conclude that Martian life did exist?
Oh, that's a very good question. It's a hard question as well. So indeed, there is the idea that Martian life has existed at some point. This comes basically from the knowing that Mars was habitable at some point. It was warm, there was a lot of water, just before the Earth became actually to be also very habitable and nice to live on. But of course, we do not have any direct evidence that there has been life. Not at all. This is only because Mars was habitable at the time point, and it's likely that there could have been life, but we don't know. There's no sign for that. There's also the idea that actually some microbes could have been spreaded from the Mars surface to Earth through panspermia before the Earth life actually evolved, but there's no proof for that. This is all hypothesis. We don't know.
Okay. So could you please explain the effect of microgravity on the virulence of microorganisms?
Yeah, that's also a complicated but good question. So we see that certain microbes, for instance, certain Salmonella strains react with higher virulence on due to microgravity. Others are reduced in virulence and have other problems or react differently. The reason for that is not really clear. Obviously, it has to do something with the fluctuation of nutrients in the medium, but also in the cell itself, which puts the microbial cell under stress. So this has to do with gradients and osmolarity as well. Obviously, there was only one paper on that, and it's not really clear why this is the case. I feel this is completely understudied, and we need to do much more research on that, because this is important. We need to know how microbes react to the space situation.
Okay, thank you, ma'am. So, what other planets do you think have the most potential for microbial growth?
That's a very good question. I think I mentioned the candidates when I showed the slide on Mars and Saturn and Jupiter. And these are the ones that are most important at the moment to analyze, because all of them bear water or ice. And well, when we look for life, we always have only one example that we can use, which is the terrestrial life. And so we always look for liquid water, which we think is important. We also look for certain carbon molecules that we know that life would need. But if this is really true, and if this is mandatory for extraterrestrial life, we have no idea. So but based on our human view on extraterrestrial life, we look particularly in these environments that I mentioned before.
Okay, ma'am. Which chemical agent is best for cleaning surfaces in space?
That's a good question. Well, basically, as I mentioned, they use the same disinfectants and cleaning agents that we also use on ground. What we saw actually in some of the clean room studies is that microbes really adapt to the cleaning agent. So if you always use the same cleaning agent at home, they also get adapted to it. They won't really harm from that. So what we implemented in the clean rooms is basically to change the cleaning agents. So for instance, use alkaline cleaners and low pH cleaners alternatively. So you're making sure that you're controlling the microbes even more down. But this is not yet done on the International Space Station. So I cannot say which is the best. It's the same as on the ground.
Okay. So there is a question. I saw in NASA an unexplained file that Staphylococcus aureus is found in one of the Apollo mission, which was coming from Moon. So how it can survive there on the Moon surface?
Very good question. I also find this story very, very intriguing. And as I mentioned before about the Martian soil, it's the same here. So as soon as you have a certain shielding against the radiation, microbes will survive that. It's not a problem for them to lie around and to wait for better times. They are not growing, they are not proliferating, they are not getting more and more microbes. They are just lying there and surviving, which is a fantastic characteristics of microbes, because they can survive in time and space, and as long as they are shielded against that radiation. And this is the case as soon as you have a little bit of an encasement or covering by soil. That's not a big deal.
Okay. So there is one more question. And does antibiotic resistance of bacteria change in space?
It does. It does. The microbes do different things. They grow differently. They react to different environments in a different way. In general, the antibiotics resistance capacity is given in their genetic code. So this is not something they can really change, but they can react faster or acquire things quicker than they might be able to do on ground. This is still also understudied, so we have to look into this further. But due to the different changed virulence, also due to the changed efficacy of antibiotics, we need to learn more about that. I agree, good question.
Ma'am, what do you think can the conditions affect the rate of evolution of life in space, and how?
The conditions affect the rate of evolution of life in space, and how? I'm not really sure if I completely understand, but you are saying basically that the evolution could be changed by spaceflight or external conditions. Yeah, because there's a different selection pressure, and the selection pressure is everything that we are talking about in microbiology in general. So microbes are very good in adapting to new situations. And of course, if there is a new condition given to them, they adapt quickly and they change. And yeah, that's just certainly would be a boost forward due to the selection pressure.
There's a question. I saw an astrobiologist. He tells that universe produce microbes on its own, but they suddenly die. Can you put some light on this?
I'm not aware of this hypothesis, but based on our knowledge, of course, we know that life comes from other life, so it's not something that actually creates by itself quickly, unless we have billions of years of time. Then we might observe a second evolution or a second development of life. But in this case, it's not very likely to have a quick production of new life out of nothing, I would say.
Okay, so here we have these questions in chat box, and now I would request other participants or audience, if they have certain questions or any query, please they can have discussion with Dr. Christine.
Okay, I have a question now, Dr. Christine. This is in context of forward and that reverse contamination which you were talking about. I do understand the problem with the backward contamination, because we don't want even more powerful microbes coming back to us. We're still struggling with the pathogenic microbes itself here, and we don't want to make conditions worse. But when we talk about the forward contamination, carrying microbes from our surface to the other planets, uh, what could be the problems with it? One is, of course, carrying them back. But if we keep that part aside, we are leaving microbes there, and we do understand that they do have a very good capability to adjust sooner or later. We have few numbers which can adapt to it. There's a possibility. So, what is that on a larger picture you think could be a problem? Let it be there. Maybe they can adapt themselves. Maybe they can initiate a life. I mean, just saying. What are your views on that?
It's a fantastic question, and it's actually very important to really think about this and also be a bit critical what makes it all sense, because as I mentioned, it costs a lot. So if you send a spacecraft, you have to do thousands of different microbiology experiments to make sure that the contamination is low. So why are we doing that? Well, there's two things. The most obvious thing for me is basically that there is a kind of an ethical issue. So we are entering maybe a new biotope, which is its indigenous biosphere. And we are bringing our own life there, and maybe our own life from Earth is so much capable of growing there and proliferating there. So we are destroying another planet with its own biosphere. So this is an ethical issue. Maybe we don't want to do that. But on the other hand, there is the problem of the mission success. And this is the basic problem behind. So if we want to find life, it doesn't make sense to bring our own life with us, because the instruments are very sensitive, and they will immediately find, let's say, the bacterial lipids or whatever that we bring with us, before they will find another life that is actually very much spread and not very abundant in the soil of Mars, for instance. So it makes your mission fail if you bring too much of a contamination. At some point, maybe when we have found out, as we did for Moon, our Earth moon, that Mars maybe is sterile as well, so that maybe there is no life, then we don't have to care. Maybe it's even thinking about terraforming, bringing specific microbes there to make it another Earth life. So that would be another question. But for now, I think as long as we have not scientifically answered the question whether Mars has life or not, we have to stick to this rule and make sure that the contamination is not too high so that we cannot find life at all there. Does this help?
Yeah, definitely. But my question was like, one thing is that we don't want to, let's say, disturb that incubator, which might be, helping life to sustain there or develop them. That is one thing. But on the larger aspect, our own intentions is to find a place where we can also go and stay.
Yeah.
Right? So why not to have life, develop there? That was the question. Of course, ethical issues are there. We should have our contamination restricted to ourselves. That is also true. But that was on a different aspect, I guess, was my question. So
it will come, I'm sure. In a few years, maybe we have answered the question about life on Mars. Then the next step, as I said, is maybe terraforming, maybe letting microbes grow there and see. It's also kind of an experiment, right? For us as a microbiologist, it would be very interesting to see what happens to those microbes in maybe thousands of years. And yeah, I would be in. So this experiment sounds fantastic, but yeah. Let's save the extraterrestrial life first and then spread our microbes.
yeah, well said. Thank you, Dr. Christine.
You're welcome.
Manpreet, do we have other questions?
Yes, I have one more in chat box. So this is, ma'am, what is your point of view about panspermia theory?
That's a very good question as well. Panspermia actually means that microbes enclosed in a meteorite or some similar stuff has been transported from a planet to another, and that might be the source of life on this planet. So let me start from the beginning. There have been a lot of different studies looking at whether microbes could survive in a meteorite. Which also looked at the impact and the type of stone you're using, how much you have to enclose the microbes into the meteorite material, and it was shown that this is possible. So microbes enclosed in a meteorite can survive the travel through space, and they can also survive the impact on another planet. So this has been shown. So in theory, this is possible. And as I mentioned before, for me, it's very, very intriguing to understand or to think about the idea that Mars has been habitable at some point and it was liquid and warm just before Earth life started. And there's actually quite a lot of numbers of researchers that think it's possible that life on Earth actually stems from Mars and has been transported through panspermia. So I think it's very likely, or it's possible that this has happened, but I'm not sure. Of course, we will never find out, or maybe not find out if this had really taken place.
So, well, we have no other question in chat box, and if
I would. May I have another question?
Dr. Tony. Yeah,
Dr.
Tony. So I think we have no more questions in chat box.
I have, if Dr. Christine, if you allow, I have one quick question. We have our students with us today, and many of them are very excited about this field, but they really don't know how to pursue in the field. So maybe if you can, guide them what to follow and how to follow if they want to pursue their career in space microbiology. That would be great.
I do not have much experience how to do it. I know only how I did it. So, and maybe some of my colleagues. And the interest, of course, is the first step. So it's very important to be interested. And then, like I did, I can tell you that internships are very important. So maybe you look at people doing such stuff for space agencies, or like NASA people, and also JAXA and so on, and many other agencies are actually also doing microbiology in a large extent. So the first step would be making contact to them and trying to do internships there. and to get into contact with those people. There's also conferences on space microbiology, at least partially in the COSPAR conference, which you can visit and you get in contact. There's also astrobiology networks all over Earth, so you can try to also contact those people. So if you have specific questions, you can also request or write me an email. I try to help, but I think making internships somewhere, it really helps to get into contact with those people. Just ask them. All of the space microbiology people are really, really nice, and they're always very helpful. But be aware, it's a little bit of a difficult field to get funding for. Maybe it's different in India, I don't know. But the funding for me was always a bit difficult to be able to work in this field.
Thank you very much for answering the question.
Thank you.
Manpreet, I guess all questions are picked up. You're on mute, Manpreet. Unmute yourself.
Okay, so yes, all questions have been picked up. So we can.
Alright, so I have one request to make to everybody. We would like to have a virtual group photo. So if you all can please switch on your camera.
Very nice idea.
Mansi, are we ready? I'll on the count of 3, you can click the photograph. Very nice.
Great to see you all.
A few seconds more, people are still coming on their camera. Mansi, let me know when you are ready.
Yes, ma'am, I'm ready.
All right, so I hope you can see a lot of faces on your screen. Please set up the layout accordingly.
Ma'am, I've taken a few screens.
Oh, you've taken already. Okay. Okay, thank you, Mansi. Now, I would like to thank Dr. Christine for joining us today, and it was actually a great presentation. On behalf of entire organizing team and everyone here today, I would like to express a sincere thanks to you. It was a wonderful and excellent session with interesting content highlighting our intense relationship with microbes. Also, the microbes, what I understood, of course, everybody knows that being microbiologists, that they're integral part of our existence on Earth to an extent that the impact cannot be ignored even while we are exploring or understanding life on other planets. So, on a bigger picture, this association, whether it is positive or not so positive, humans and microbes, they still belong to each other. So, your talk was one of the most engaging I've ever had the privilege to listen to. Thank you so much for that. And heartfelt thanks for generously accommodating our invitation to speak in your busy schedule. So, we are grateful for your time and effort you took to share your insights on such an intriguing topic. And I would like to mention here that a token of gratitude from our side, we have sent you a cute little potted plant already, which is on its way to you. And I guess it's no longer a surprise. You might have, been receiving some kind of intimation for that. So I very humbly request you to accept that. So once again, thank you for joining us, and it's been a great, great pleasure and privilege to have you. And looking forward to having you in future too. Thank you so much.
Thank you very much. Thank you for inviting me. It was a big honor to be with you. I hope you still enjoy the rest of the colloquium. So good success to all contributors and organizers. Stay healthy and take care. Hope to see you soon.
See you soon. Thank you so much. Thank you.
Thank you so much.
All right, now moving ahead, we have our second speaker for the day, Dr. Tony Gutierrez. And now let's get ready for another interesting session. I would now request and invite Dr. Anita Kapila to introduce our speaker to the audience now. Dr. Anita Kapila.
Thank you, Kruti. I hope I'm audible.
Yes, ma'am, you are.
Okay. Well, joining us today is Dr. Tony Gutierrez, who is Associate Professor in Environmental Microbiology and Biotechnology at the Heriot-Watt University, United Kingdom. Dr. Gutierrez is a modern-day legend with an ever-expanding interface of research activities on oil-degrading bacteria and their roles in biodegradation of oil spills, as well as bioprospecting marine microbes for production of biosurfactants and bioemulsifiers. Dr. Gutierrez, after completing his PhD in microbiology and immunology at the University of New South Wales, Sydney, worked as a postdoctoral fellow at the University of Florida, USA, and the Scottish Association for Marine Science, Scotland. In 2008, he received the Marie Curie Research Fellowship, which took him to the University of North Carolina, where his work was on oil-degrading bacteria in the oceans. In 2012, he joined the Heriot-Watt University as associate professor, and his lab works on microbial responses to oil spills, fate of plastics on land and in oceans, bioremediation, and microbial biotechnology. He has published more than 80 research articles in peer-reviewed journals. He's been the reviewer for many journals, including PNAS, and has many undergraduate, postgraduate, and PhD projects under his supervision. More importantly, he has been actively involved in raising awareness about the impact of microbes on the quality of our lives, and would life really exist without them? I now invite Dr. Gutierrez for sharing his thoughts, findings, and his observations. Welcome, sir, and over to you, sir, Dr. Tony Gutierrez.
Thank you very much for that very kind introduction. I don't believe I'm a legend, but if you say so, okay. You can all hear me, right? So I'm just going to share my screen of my talk. Okay, can you all see that? My screen there?
Yes, sir.
Excellent. Okay. Great. Okay, so first of all, hello, and I want to start with thanking the organizers, thanking you for inviting me to give a talk as part of this 3-day colloquium event. I'm Dr. Tony Gutierrez. I'm an associate professor at Heriot-Watt University in Edinburgh in Scotland, and it gives me a lot of pleasure to talk on environmental microbiology and the great good that microbes do for us and to our planet at large. So just moving on to the next slide, just making sure I can do this. Here we are. There's the button there. Right. So just an overview of my talk today. I'm just going to begin with a very brief overview of microbes in our lives from symbiosis of sustaining life on our planet. And then I'm going to move into one of my research loves, which is in the role of microorganisms in oil spills. I do a lot of other types of research, microbes involved in plastics degradation. Of course, that has become a very fashionable and important challenge today, which is the plastics pollution. I also work on natural products discovery from microbes, particularly in biosurfactants and their uses in different applications. And then I'm going to just end this talk with the role of microbes in combating oil pollution. And I'm going to focus a large part of my talk on the Deepwater Horizon oil spill, which was a historic spill. I just wanted to mention that I do have a TEDx talk. I know that one of the organizers mentioned to me that they picked up on that, and a reason why they contacted me to give a talk here. In that talk, actually, I give an overview on the intricate relationship of microorganisms with humans and with different types of animals as well. So if you're interested in that, by all means, please go to that site. It's a YouTube TEDx talk there that's available online. So today I'm just going to give a little introduction on what I covered in my TEDx talk, but I'm going to then focus in on the Deepwater Horizon spill. So I want to begin here to describe the close relationship we have with microbes. We can look at the miracle of life here, which is when one fertilized human egg cell becomes 10 trillion human cells that make up each and every one of us. We are on average about 10 trillion human cells, but each one of us also contains, on average, 100 trillion microbes. So we are a living ecosystem made up of what are human cells, but also 10 times more microorganisms. Our bodies contain about 10,000 or so species of microbes. So this is effectively our microbiome. And by numbers, we are actually more microbe than human. And there's a lot of interest today, a lot of interest that's increased over the years in terms of the gut microbiota in diseases, in health. So there's a lot of work that's involved in that. Now, I'm not an expert on that, and I'm not going to be discussing that today, but there's certainly a lot of very interesting work on that. And still, it's taking the medical establishment time to actually capture that and take that in as something that they would consider in terms of fighting disease. But it's taking time, but I hope eventually the gut microbiome will become an established medicine in science. So moving ahead. So yes, just showing you here how we have microbes in different parts of our bodies. Of course, they're all over our skin. They're in the urogenital tract, gastrointestine, of course, huge amounts. They're in our mouth and also our noses as well, and our ears. Right. I also often like to start by giving a very basic idea of how small microorganisms are, and balance that against how incredibly impactful they are upon our lives and that to all other life on our planet. You could quite easily accommodate about 1 million bacterial cells on the tip of a pinhead. A dozen or more would line up closely on the filament of a hair, like so. So it just goes to show how tiny these things are, yet how important they are to life on our planet and our close relationship with them. So I'm just going to get into that now. So what if we had a world without microbes? I like to start off with. So what if we had a world without bacteria at the top here, algae, no yeast? Well, we wouldn't have diseases, which is a great thing. We wouldn't have food poisoning, and we wouldn't have these pandemics and horrible diseases and like Ebola and the current pandemic that we're having with COVID-19. We would also not have food spoilage. We wouldn't have bad body odor, and there wouldn't be fish kills from eutrophication in lakes and rivers. So that's all great, but things we'd miss out from not having microbes. Some of the things we love, like for example, yogurt, beer, wine, and other types of beverages, bread as well, cheeses, vinegar. And this is just a sample, just a small sample of different types of foods and amenities and so forth that microbes are so important in producing for us and things that we love. So we're missing out on that. But there's also one more thing we'd be missing out on. Well, I'm not sure we'd be missing out on, but the thing is we wouldn't have microbial decomposition. So when we die, when animals die, anything living dies, if there were no microbes, animals, dead animals will simply accumulate. They wouldn't decompose. Chemically they would, but it would be extremely slowly. So we need microbes to decompose things and for organic matter to continuously be recycled. This is life. This is just natural, beautiful life. and it wouldn't be without microbes. One other very important thing, type of microorganism is what we find in our oceans, and these are the phytoplankton. These organisms are essentially producing the oxygen in every second breath that we take. So if you look here, all the green patches that you see here, all the green patches in our oceans of different various intensities, those represent, from satellite imagery, phytoplankton blooms in our oceans. And these phytoplankton blooms are essentially the rainforests of our ocean. Some people say it's the second lung. These organisms, simply a microscopic algae, producing 50% of the oxygen in our atmosphere. And they also play other very fundamental important roles, like in the biological pump, taking carbon dioxide from the atmosphere, incorporating that into carbon biomass, and then that sinks down to the ocean floor. Some of it gets deposited into sediment, so it's the carbon pump, carbon sink. Some of it gets recycled as part of the microbial loop in the water column. Very important to generate nutrients and energy and carbon flow in our oceans. So it just goes to show, these tiny little things are so fundamentally important in sustaining life on our planet. Now, all living things, as I've mentioned, have some relationship with microbes, and I'm just going to give you a small subset of some of the intricate relationships that microbes have with various animals. To start off with, the gut microbiota in us as humans, but also animals as well, very important in terms of digesting foods, absorption of nutrients, in interaction with the immune system, as well as scientists have shown that gut microbes have some sort of influence on the way we think sometimes, or our emotions. Not so much the way we think, sorry, but more of our emotions. Moving along, this is the hoopoe bird. And this bird paints its eggs. Here are the eggs, it's sort of slightly blue. It paints its eggs with bacteria-rich fluid that it secretes from a gland beneath its tail. The bacteria release antibiotics that stop bacteria from infiltrating the eggs, and which could cause harm to the chicks. So an intricate relationship there between the bacteria and the bird through its eggs. Here we have the antlion. This is a larval beetle with ferocious jaws that use bacteria to produce toxins that paralyze its victims. Here we have the Colorado potato beetle that uses bacteria in its saliva that it secretes to suppress the defenses of plants that it eats. Here we have the spiky pufferfish uses bacteria to make tetrodotoxin that poisons any predator that tries to eat it. Here we have the anglerfish, uses luminescent bacteria to attract its prey. Here we have the cardinal fish that houses luminescent bacteria to attract its prey. And here we have coral, and inside the coral, in its polyps here, you can see the little green bits here. These are zooxanthellae. These are microscopic algae that are found in these coral polyps, and they are important in producing food for them via photosynthesis. Without the microalgae, the corals would bleach and die. And one last animal microbe relationship that I want to present to you before I move on to the next chapter of my talk. And that is to do with the bobtail squid. This is probably one of the cutest little squids in the sea, but there is much more to this squid that's crucial to its survival. And it has a lot to do with a symbiotic relationship that it has with a specific type of bacteria. So bobtail squid have a light organ inside its mantle here. This light organ is fueled by a symbiotic bioluminescent bacteria called Vibrio fischeri. Now the light organ, which is butterfly shaped, is situated in the mantle cavity and is used to cancel out the bobtail squid's silhouette. So if you look here in the water column, okay, in the water, there's the bobtail squid. It produces this light in its mantle, and it shows this light like a flashlight. Okay, this is just representing the light shining down. And by the full moon, this is when these squid come out, by the full moon, this light cancels out the light from the moon. So the silhouette of the bobtail squid is cancelled out. So therefore, any predators underneath the squid will not be able to see the squid. It's very similar. You know, it's been described very similar to, if any of you have watched the Star Trek, it's like the Klingons. You know, they turn on their vanishing shield. You know, they makes their ship disappear, and you can't see it. It's very similar to that. So it's like a Klingon technology that this bobtail squid has. Now, if you were to take some seawater into a glass, hold it up to the light and take a good look, apart from seeing possibly a few floating or suspended specks, it would appear as a clear liquid. If you drop it into a microscope and add a little bit of a stain, you'll see a whole universe in there. I mean, typically in a liter of seawater, you'll find about 10 billion viruses. You'll find about 1 billion bacteria, and you'll find hundreds of thousands of microscopic living plants called the microalgae, the phytoplankton. So microorganisms in the ocean perform some of the most important functions in sustaining life as we know it on Earth. They are also the key to the origins of evolution of life, and possibly could play a similar role in other planets where life may exist too. Microbes are important in decomposition and the recycling of nutrients, as I mentioned before. But there's a particular microbe in the oceans that's important in keeping the oceans clean from pollution, a particular type of pollution, oil, crude oil. So let's have a look at that. So this slide here shows you the number of oil spills, large oil spills, greater than 700 tons from 1970 to 2013. An important message from this slide is that oil spills at sea over time have decreased. Unfortunately, this is due to more health and safety. Ships are built strong. Oil tankers are built with double hulls. So if a ship runs aground, it may rupture the first exterior hull, but not the internal one. And there's a lot more health and safety. So thankfully, spills have decreased over the years. And you can tell here by the droplets here as well, the volume of oil that spilt into our oceans in the 70s, early 80s was much greater than what it is now. And I wanted to show you this slide just to show you that oil in our oceans doesn't just come from human accidents, like oil tanker spills, accidental spillages and all that sort of thing. So this side of the pie chart on the right-hand side shows you the proportions of oil that spill in our oceans every year from human activities. And the left side of this pie chart shows you the proportion of oil, which is about 49% of oil that spills into our oceans from natural oil seeps. So this is oil that seeps into our oceans every year naturally. And it's been doing so for thousands upon thousands of years. So this is from underground oil reserves just naturally seeping out. You have a lot of these oil seeps in the Gulf of Mexico, for example, in the Gulf of California as well, the Guamas Basin. So the message from that slide is hundreds of millions of gallons of oil enter the oceans every single year. Our seas and coastlines are swamped with oil. I mean, you go to the coastline, you don't see oil slicks there. In some places where there's been a recent oil spill, maybe, yes, of course. But generally speaking, if I were to step out on the coast here of Scotland and look out into the ocean, I do not see black crude oil. So why is that? It's essentially because of these organisms, these microbes. These are the ocean's gas guzzling machines. They just love oil. Some of these organisms just eat oil. They're called obligate oil degraders or obligate hydrocarbon degrading bacteria, and they devour oil. If you give them sugars to eat, other carbon sources, they will not touch it, but oil they will devour. And there are other types of oil degrading bacteria that are referred to as generalist oil degraders. That means if you give them oil, they'll eat it. If you give them sugars or other carbon sources, they'll devour that as well. So they have a more versatile ability to break down and use other types of food sources for growth. Now, we've come a long way over the years since the first isolation of the first oil degrading bacterium, which was a rhodococcus, previously wrongly named as Mycobacterium. That was back in 1913 by Songen. Now, this phylogenetic tree here just shows you the relatedness of the different types of oil degrading bacteria that we know of today. But this phylogenetic tree, I've highlighted in colors the obligate oil degrading bacteria. And here we have Microbiome alkanoclasticum up there. That's a gram-positive organism. All the rest are gram-negative. So here we have organisms of the genera Oleophilus, Oleospira, Thalassolitis, Alkanovorax, Cycloclasticus. These are your typical obligate oil degrading bacteria. That means they absolutely love oil. And you give them something else to eat, and they won't eat it, unless it's, of course, metabolites of central metabolism like fumarate or lactate and things like that they might eat. And in terms of the distribution of these obligate oil degraders in their oceans, they're practically everywhere. So this map is a little outdated, but it shows you all these dots represent where obligate oil degrading bacteria have been isolated from. And as you can see, you find them mainly on coastlines because that's where most research cruisers go to because it's accessible, coastlines or slightly offshore. Very rarely do you find research cruisers going really right out into the middle of the ocean because they're more expensive, these research cruisers. But if you were to, the fact of the matter is, these obligate oil degrading bacteria are ubiquitous. They're found all over the world. Now, as part of my Marie Curie fellowship when I was in the United States, I was at the University of North Carolina, Chapel Hill. I was looking to the discovery of new types of obligate oil degrading bacteria, also generalist oil degraders as well, from microscopic algae, so from phytoplankton. Now, phytoplankton, as I've shown you, are very important in terms of producing 50% of oxygen in our atmosphere. They do all sorts of different important functions. So one thing from my work, and I worked with a colleague here in Scotland on this, Dr. David Green, who started this. We looked at isolating these oil degrading bacteria from microscopic algae. And the hypothesis and the idea was that these microscopic algae, here showing you phytocystis cells, are coated with hydrocarbons because phytoplankton are known to produce hydrocarbons. And so you would suspect these oil degrading bacteria to be living with microscopic algae, and thus they are. We isolated many different types of oil degrading bacteria. And a very simple experiment to do is simply take microscopic algae in a flask, add that to a bacterial medium that contains a particular hydrocarbon like phenanthrene, naphthalene, or hexadecane, incubate it, and then over time you'll often see a color change, which is the production of metabolites from hydrocarbon degradation. You plate it out, and you can isolate these sort of hydrocarbon degrading bacteria. And this is showing you again a phylogenetic tree of the different types of obligate oil degrading bacteria on a radial phylogenetic tree here, so it's circular, showing you the same names. Remember Thalassolitis, Holospira, Alcanovorax, Holophilus, Marinobacter, Cycloclasticus, and we discovered new types of obligate oil degraders that we could add to this tree, and which I got to name, which is a lot of fun naming these organisms using Greek and Latin terms. This one we're called Porticoccus hydrocarbonoclasticus. As you can see, the species epithane name suggests it's a hydrocarbon degrader. Here, another one, Algophilus aromatissivorans, breaks down aromatics, and Polycyclovorans algicola, again, breaks down polycyclic aromatic compounds. And these are just some photos that I took using the electron microscope scanning or transmission. And they're just typical looking bacteria. It's what they do that's incredible, but they just typically do look like any other cell under the microscope, really. I wanted to show you this because this shows you one of the other types. This was a generalist hydrocarbon degrader that I isolated, published in this paper here, and I show here an image. This is actually an oil droplet, crude oil droplet, and it's completely covered in these bacteria. So they just absolutely love to attach to oil and eat it up. They love to devour it. In my group, we developed fluorescence in situ hybridization probes, so they're called fish probes, to target and detect these types of bacteria directly on phytoplankton cells. So here's an image of the bacteria we're targeting. And then we take phytoplankton, like these diatoms here, and we apply the probes, and we're able to observe. So these bright colors, these bright tiny little purple signals are the bacteria living on these pinnate diatoms here. So it's very nice to see them, living in association with the phytoplankton. And here's an image of a paper that we just published a couple of months ago in the journal Microorganisms. And this is Polycyclovorans algicola, obligate degrader, these tiny little dots they're bright orange, living associated with these chains of skeletonema. So this is the diatom under the microscope, and under the fluorescence the fluorescence microscope, after applying the probes, you can see these bacteria attached directly on these diatom cells. And so here is the paper that just came out on that, where we also applied flow cytometry coupled with fluorescence in situ hybridization to directly show that these bacteria are attached very tightly and closely attached to phytoplankton cells. And we showed this for a particular size of phytoplankton in the ocean, of 50 to 70 micrometer cell size population of phytoplankton, from field samples that we took in the Northeast Atlantic. So this was not using laboratory cultures of phytoplankton. We actually decided to go on research crews and take samples and directly interrogate those samples to see if we detected these obligate hydrocarbon degrading bacteria associated with the phytoplankton, and we did. And the journal editors, they found it, I guess they liked it. They put the image on the cover of the issue, which was quite nice, I thought. So in summary of this work, some of the questions we ask is, why are these specialist oil degrading bacteria found living associated with eukaryotic phytoplankton? Why are they living together? Could it be to do with adsorption and accumulation of hydrocarbons on the actual microalgal cells that attracts these bacteria to attach and live associated with them, because that's where their food source is, the oils? Could it be some sort of commensal or mutual sharing of carbon or some nutrients between the bacteria and the algae, as is found with other types of bacteria and algae, in terms of sharing vitamin B12, in terms of producing siderophores for iron capture and making iron more bioavailable to the algae? Or could it be to do with, it's just the surface of the microalgae? It's just possibly just a nice place to live and hang around. So a lot further work needs to be done on this. It's certainly in a nascent stage of understanding. So now I'd like to move on to the Deepwater Horizon oil spill. which I did a lot of work on when I was in the United States. There in 2010 it happened. I was there and I got involved in a lot of work to investigate this oil spill. And it was that big. They made a movie out of it starring Mark Wahlberg and Kate Hudson. Now, this is just a depiction of the Gulf of Mexico spill. And as you can see here, so here's the burning rig, which eventually broke off. The pipe from the rig down to the seafloor completely broke off as well and collapsed. And so oil on the seafloor here was just continuously gushing out. Many gallons, I mean, in hundreds to thousands of gallons per day, just oil gushing out for a period of over 60 days, about almost 2.5, 3 months. So, you know, it's a historic oil spill. It was massive. The one that compares to that in terms of how huge it was, in terms of volume, was the one that occurred in the Bay of Campeche, also in the Gulf of Mexico, but in Mexican waters. And that was the Ixtoc 1 oil spill. That was also one that occurred, continuously gushing of oil into the Gulf for almost a year. And it was in shallower water, though, in about 50 meters of water, whereas this Gulf of Mexico oil spill occurred in 1.5 kilometers depth of water. And so oil just gushed out. A lot of it reached the sea surface, and quite a lot of it also formed this oil plume. It's basically a cloud of oil that's dispersed and solubilized oil that formed at about 1,000 to 1,200 meters depth from the sea surface. And it just moved like a cloud in a southwesterly direction. And this picture just shows you, the different strategies that were used to combat the oil spill. There was some burning, there was spraying of dispersants. There was also spraying of dispersants directly on the broken riser pipe at the bottom here. And also ships and so forth that were trying to work on capping the leaky well. But at the end of the day, the best arsenal used to combat an oil spill are just the natural communities of oil degrading bacteria. So here's another depiction of the oil spill in the water column. Various researchers' crew. So first of all, here's the water column from sea surface 0 meters down to 1,500 meters depth, and this is the timeline from March when it before the spill to April when it occurred. April 15 was when the disaster, the blowout occurred, and it occurred all the way into June, July. June, July here. This is after the oil spill over here. And myself and colleagues, we worked on samples that we got from research cruises. Many other research groups around in the United States and the world worked on this as well. And we reported, and others as well, that there was a very strong response from an organism called Oceanospiralalis within the oil plume that formed at depth. On the sea surface, there was a very strong response of Cycloclasticus, which is an organism that degrades aromatic compounds, preferably in the oil. Oceanospiralalis had a preference for both, but mainly the aliphatic, long-chain hydrocarbons. And we also saw an enrichment of Cycloclasticus and also Colwellia. And further research on Colwellia showed that it possibly had a role in breaking down and degrading the dispersant that was applied to combat the oil spill. And so this just shows you here in terms of the microbial response demonstrated with pie charts. Here showing you the green here, a very strong enrichment of Cycloclasticus compared to the total microbial community there. And an enrichment also here, shown by the orange, of Halomonas, Alteramonas, and Pseudoalteramonas, which are organisms that are commonly that are known for degrading oils as well, but also producing exopolysaccharides, so polymers that they release. And we'll get to that in a moment. Above the plume, so there was an oil plume that formed at this depth here. Above the plume, there was a higher microbial diversity, as you can imagine, as you can expect, because there's less oil in that region. And in the plume, there was a very strong enrichment of that Oceania spirulalis. And below the plume, where there is less oil, just under it, there was a higher diversity of bacteria, as you would expect. And these are just pictures of the Cycloclasticus. These tiny little green dots surrounding oil droplets. They just love the oil. They just attach to it and try to devour it. Now, one technique that I used during the oil spill was a stable isotope probing. So this technique was using 13C labeled aromatic hydrocarbons or aliphatic hydrocarbons. But the important thing is that the carbons on the compound are 13C labeled. So the idea is they're 13C labeled substrates that you add, and over time, the microorganisms that can break those and utilize those the carbons from that hydrocarbon incorporate them into the biomass. So in making of cell membranes, cell walls, and importantly, DNA, because that's what we want to recover, the DNA, because it's the DNA that tells us what type of bacteria they are that are breaking down the hydrocarbons, because we use the 16S gene marker as a taxonomic marker to identify who they are. So the idea is you incubate your seawater from the Gulf of Mexico, in this case, in a flask, together with the substrate that's labeled 13C, and over time, the microorganisms that can break down and utilize that hydrocarbon will incorporate that carbon into their DNA, and that's represented here by the red organisms, and the ones in blue are those other organisms that do not degrade or touch that hydrocarbon substrate. And then we take all that DNA, we extract it, and we separate it using a gradient of cesium chloride that's spun very fast, creating a gradient that separates the light DNA that's not labeled compared to the 13C labeled DNA. We then take that 13C labeled DNA that represents the community of microorganisms that broke down and degraded that 13C labeled substrate. And we do a lot of bioinformatics analysis with it. We identify what those bacteria are, and we can reconstruct genomes and determine what other metabolic functions they do and so forth. So we can certainly do a lot with that. And so we published a paper on this in Nature Microbiology, and it shows you just here's a representation of the spill. Here, there's the leaky well, there's the oil plume, and we took samples from the sea surface and from the plume, and we incubated them with a 13C labeled substrate. I used hexadecane, and I also used in other experiments phenanthrene, and in another one naphthalene. That was incubated. We took extracted DNA, separated out the DNA, the light from the heavy. We're most interested in the heavy, and we did shotgun metagenomics on it. And eventually we reconstructed the genomes, and we were able to determine the pathways of degradation that microbial community has, and we can determine, what they were doing during the spill, what genes they had in breaking down the hydrocarbons. And what we show here is the pathways that we were able to reconstruct. And here we show that we identified Marinobacter in that heavy DNA, 13C labeled DNA community. We identified Thalassospira, Alcanovorax, Alteramonas, Cycloclasticus, Neptunibacter, and Colwellia. And beside here, we show which incubations we identified these organisms from. So Marinobacter in our incubations was degrading hexadecane. These organisms here were degrading naphthalene, and these here phenanthrene. And we were able to, from the reconstructed genomes of these organisms, we were able to identify and reconstruct the pathways for degrading these hydrocarbons. So here we have the pathway for degradation of naphthalene. Here we have the pathway for degradation of phenanthrene. I'm not showing you the one for hexadecane. It's too much to show on a slide. So I'm just showing the polyaromatic hydrocarbons here. And you can see that not all of these bacteria contain all the genes required for degrading naphthalene or for degrading phenanthrene. So some of the genes are there in Marinobacter, and some of the other genes there are in Thalassospira or Alcanovorax or Neptunibacter. So the thing that what we found from here is that it's no single organism that's a key to breaking down hydrocarbons. These organisms behave, or their role is as a network. They work as a community. One of them does this, the next one does the next step, the next one does another step, so they work together. And that's the beauty of these microorganisms in getting rid of the oil, in combating the oil in the Gulf of Mexico. They work as a community. Now, another part of my work was that we investigated was the Mosfet event. So this was marine oil snow sedimentation and flocculent accumulation event. And this was basically, it was unprecedented. It had never been recorded before for a major oil spill at sea. And that was where large amounts of polymer, mucus-like material formed on the sea surface when the oil spill occurred. And this eventually sedimented down to the sea floor. And this photo image here, taken by Arnie Dirks, shows you the sea floor and these polymers just sedimenting down like snowflakes to the sea bottom. So some of us researchers started to focus in on this, what was happening, why. Here it shows you an image here of these mucus-like aggregates on the sea surface. This is a picture in the Gulf of Mexico. And under the microscope, I used particular techniques to capture this. And here it shows you under the microscope, little tiny oil droplets in seawater. And here are these aggregates, these cloud formations. Now this image, if you look at it for the first time and you didn't know, you'd think it was a galaxy. It's just amazing, but it's actually just oil droplets and these mucus-like material that bacteria and phytoplankton produce, and it seems to capture the oil droplets within it. We did some, before I go into what this means, so this just shows you some more photos on the sea surface in the Gulf of Mexico of this mucus stuff. And this is the marine oil snow, that we call it, being formed in the laboratory in cultures. And this is by my colleague, Kaiser Vogel, that took this picture. So I started to do some experiments on this, forming marine oil snow in the laboratory. And I took a lot of pictures under the microscope. Here it shows you some of these aggregates that are formed, and those tiny little dots, bright green dots, it's jam-packed with bacteria. And also with oil droplets as well. And using differential staining techniques, Kumasi blue that stains protein, and using Alcian blue that stains polysaccharide, I was able to show that, and my students as well, that these aggregates are made up of polysaccharide and protein, but they're jam-packed. They're hot zones for microbial activity. And here it shows some more pictures here showing you just these tiny little dots that you see here are just all bacteria amongst these oil droplets. And this is a whole aggregate of this mucus stuff. And in Kaiser Vogel's work, he showed that there's a lot of microbial activity happening here. So there's a lot of lipases and enzymes being formed, quite possibly involved in breaking down the oil. In my group's work, we showed that these aggregates contained oil-degrading bacteria, things like Hylomonas, Alteromonas, Pseudo-Alteromonas, and these are involved in breaking down the oil. So it's quite possible, the hypothesis is that these marine oil snow aggregates are hotspots where oil degradation rates are happening. And we're still working on that to explore that even further. There's also a lot of work being involved in what forms this moss. We really still don't understand fundamentally what are the mechanisms involved in marine oil snow moss formation. We know it's the presence of oil. We know that you need oil to form this moss stuff. And note that moss is exactly the same as the formation of marine snow. It's important in the biological pump, but in this case, it's with oil. could it, these are just questions posed here, does it involve, is it to do with the quality and the quantity of dissolved organic matter or particulate organic matter? Is application of a dispersant, a chemical dispersant, also trigger its formation? Our experiments have shown that, but we also have experiments that in the absence of dispersant, you also get moss formation. Do mineral substances, calcium carbonate, metals and silicates, are they involved in it? So there's work, there are researchers that have proposed that. Also phytoplankton cells and the polymers they produce, or the polymers produced by bacteria as well, are these important? We believe so, because we know that these moss particles are made up of polysaccharide and protein, or glycoproteins, which is what phytoplankton and bacteria produce, polymers. That's what they're made up of. So there's a lot of questions, still research to be done on this. It's a very interesting, hot topic. So I just want to, I hope I'm on time. I just want to finish up here with just a summary, and just that microbes are essential in providing us many services and also sustaining life as we know it. So I just wanted to give you a picture, just a brief overview on that. And before I had gone into the main part of my talk, which was on oil degrading bacteria and the responses of oil degraders in combating oil spills. So huge quantities of oil enter our seas and oceans every year from both human activities and natural seeps, as I presented. But oil degrading bacteria are key to keeping our oceans in check from oil accumulating in the ocean. And many different species of bacteria can degrade oil, but it's those obligate degraders, those specialists that are so fundamental in really tackling the oil. But not to say that, sometimes generalists can also be the dominant organism in getting rid of the oil. Over and above the obligate degraders sometimes. It depends on the environment. A novel species of obligate oil degrading bacteria have been discovered from marine microalgae, and there's many more, I'm sure, that await discovery, and there's a lot of work still going into this. I'm certainly exploring this even further. And lastly, the Deepwater Horizon oil spill clearly demonstrated the role and importance of oil degrading bacteria in responding to oil contamination, and these organisms are really our natural purging key organisms in keeping our seas and oceans clean from oil. Right, and a lot of this work cannot be done with members, researchers, PhD students, postdocs, and this is just a picture. I acknowledge all my students and postdocs that have done a lot of this work during my time in Scotland. And I want to finish up with acknowledgements of different sources that have funded my work, NERC, Marie Curie, so the European Commission, Royal Society, etc., and so many people, my colleagues, researchers around the world. And I have not included everyone here, and I apologize if I've missed anyone, but I thank so many people for their collaborations, which has been absolutely made my science life so much the happier. Thank you, and I look forward to questions from the panel.
Thank you very much for a wonderful talk, Dr. Tony Gutierrez. Now, our house is open for questions. For this, I would invite our second-year student, Priyanka, to moderate this question-answer session. Priyanka?
Hello, everyone. Hello, sir. It was a great session. Thank you so much for wonderfully enlightening us on this theme. So now it's for the questions. Yes, Dr. Sunil, you can unmute yourself and ask the question.
Hello, it was a very nice talk. Thank you so much, Dr. Tony. I was just wondering, we have a lot of oil waste being generated by refineries, and can those same bacteria be used for those as we are using for the oil spills, or they have something different? Because you have to reclaim the whole soil, the bioremediation that has to be done. So can you throw some light on that?
So in terms of the oil refineries, What are you suggesting in terms of using the bacteria for what? Sorry.
So the oil refineries, they produce a lot of oil waste, and they degrade the soils around those refineries. So do you have some idea as to what microbes, what kind of bacteria you can use for that particular bioremediation? Because this is not a ocean environment, it's a different environment altogether, and you want to recover those soils which are getting which can restore the balance of the beneficial bacteria and all that.
Yes, so that's right. There's been a lot of work in terms of because there's a lot of polluted soils in Europe,
right,
in the United States. Yeah. There's thousands upon thousands, millions, in fact. And
right.
so there are a lot of techniques, in fact, to bioremediate contaminated soils that are contaminated with hydrocarbons. And there's bioaugmentation where you seed the soils with bacteria or organisms that can degrade the oil. There's also biostimulation where you add fertilizers and that can stimulate the indigenous microbial community to clean up that oil from a particular site. So one strategy could be to try and isolate the oil degrading bacteria from that contaminated soil, is to isolate them and understand you can sequence their genomes and understand, or do experiments with them to understand what could you add to them to make them degrade the oil faster, or to trigger them to break down oil hydrocarbons that are more difficult to break down, because crude oil and petroleum, it's made up of all sorts of different types of hydrocarbons. So if you can trigger them to break down hydrocarbons that are more difficult to degrade, so it's really difficult. It's a very general question, so it's very difficult to say what's going to work. It's really doing tests without that environment, trying to find out what could work best. And this is a bioremediation strategy, and there are also chemical strategies where you add chemicals and physical processes. They can be expensive. I mean, at the end of the day, a lot of researchers we do accept that natural attenuation using microorganisms in bioremediation is usually the best technique.
Right. I have another question. Sorry, can I ask? So for the Bob squid that you mentioned, the Vibrio fisheri, I read somewhere that it has to be a quorum sensing, the phenomenon by which in the night time they attain that particular quorum, and in the daytime the quorum is not there. So can you just explain that? What exactly happens?
So quorum sensing is very much to do with sending signals, chemical signals, and this occurs between a population of bacteria at a particular time, and that's the way they communicate. So that's I mean the relation. Can you imagine? I mean, for the bacteria to know when to turn on that light, and to help the squid, this relationship is just unbelievable. So there's in that cavity of the squid, the bacteria need to know when to turn on their light. So their intricate relationship, symbiosis with the surface skin, inside the cavity with the squid is so intricate. There's a chemical communication between them. And I mean, I'm not an expert on this, but I do know that, from reading up on this, that it's just so intricate that they just know when to turn on. And quorum sensing is a strategy that's involved in that.
Okay. Thank you so much, sir. Thank you.
Priyanka, I have a question. Can I ask?
Yes, sure.
Thank you, Dr. Tony, for a nice lecture. I would just like to know, go a little tangentially to what you have explained, because in the oil we have aromatic fractions as well, right? And you just showed the entire graph of how the mechanism of degradation of those aromatic hydrocarbons takes place. Now, just going to that particular point, I would like to know, there are fungal enzymes as well which degrade the aromatic fractions. Since I have worked on lignin degradation, lignin being a aromatic highly complex compound having lots of aromatic and aliphatic linkages. So there is an enzyme called lacase, which is an oxidoreductase, which works very well, you know, and occurs by free radical mechanism. It degrades those fractions. So I would like to know if there is anything, any of the fungal enzymes that can be of any use in this.
So usually fungal enzymes are more commonly play a role on land, on land. In the marine environment, there is no question that fungi have a role to play in hydrocarbon degradation. But when, if you look at the literature, you know, when you look at what dominates and what becomes enriched strongly during an oil spill, it's bacteria and particular types of bacteria. That's not to say that the fungi do not contribute to this. They have a role, and probably quite likely more in sediments. But in the ocean, certainly it's these obligate oil degraders and generalist oil degraders that are bacteria that play a dominant role. And yeah, very true. I mean, lacases are so important. There's a lot of work being done on this. Lignin degradation, and as you say, lignin is such a very solid, complex aromatic compound. It's just so complex that, you know, for enzymes to tackle it, they really need to be quite unique type of enzymes to be able to battle with lignin.
Yeah. So, I mean, in their lignin degradation, just a point to add, these enzymes are non-specific, right? So they cause the degradation by free radical mechanism, you know, oxidation reduction, and then they generate certain free radicals, oxygen radicals, and which they degrade by the compound by free radical mechanism. Any such possibility in case of the aromatic fractions of oil?
I mean, it's possible. I haven't worked on that, but one has to take into consideration that maybe these organisms, these fungi, do not function so well in a marine seawater environment. So I haven't explored that in my work, but it's something possibly to explore. I would just say that, you know, it's conditions do have a factor in what becomes enriched and what has a dominant or a key role in breaking down aromatic hydrocarbons. I'm not sure if fungi would have so much of a role in that in seawater, but in soil environment on land, yes.
Okay, thank you, Dr. Tony. Thank you.
So the next question is it possible to stimulate conditions present in ocean for culturing these bacteria in labs?
And does I mean, you can culture and cultivate these bacteria in the laboratory. Many cannot be cultured, though, of course. As we know, 1% of bacteria, land or marine environment, can be cultured in a laboratory, and just a lot of them cannot. Yeah, I don't know if I've answered the question, though. Do you mean to change the ocean environment? Is that the question? I don't know.
So actually, it's not clearly mentioned.
Okay.
So the next question is, with present-day climate change and pollution, how are their survival and processes are being affected?
How are what processes? Sorry.
Their survival and processes. Like there's a climate change going on and pollution is there. So how are their survival and processes are being affected?
Okay, so if I've captured the question correctly, so the change, what we're experiencing with climate change is you know, really significant, and it's really important we understand how climate change can impact the microbiology of the ocean, and in particular things like ocean acidification, for example, changing the pH of even by point something of a value is significant, because pH is on a logarithmic scale, and this is particularly the case with calcium carbonate producing organisms like coccolithophores, at least phytoplankton, corals as well. So if pH goes down and coccolithophores are going to have a problem, dealing with that, then of course think about it. The bacteria living with them are going to have issues as well. Or over time, because climate change and ocean acidification happens over a lot, not just in a year, it happens over decades and so forth. Maybe these organisms will evolve over time to live with a slightly more lower pH environment. Change in temperature of the global ocean could impact as well. I mean, think about possible phytoplankton blooms that produce neurotoxins and that sort of thing are probably going to be more prevalent in the tropics. The Arctic is going to be interesting to see what happens with that. And in terms, bringing it all back to oil degrading bacteria. So I have a couple of papers that are going to be coming out this year, I hope, where I had a PhD student who was working specifically on this, looking at ocean acidification conditions to responses by oil degrading bacteria. So we're going to hopefully publish some of this work this year, and we have seen that there are differences. So change in climate, climate change can have an impact on the response of oil degrading bacteria to an oil spill.
Okay, so the next question is, have you tried bioaugmentation in oil spill degradation?
No. So I'm not very much in favor of it. So in the literature, there are studies that have come out saying that bioaugmentation worked, and there's a lot of literature that says it doesn't work. I'm very much more in favor that let the indigenous organisms do the trick. The oil degrading bacteria are practically everywhere, and they will respond to an oil spillage. It's more, I'm more inclined to add something to help them, like more of a biostimulation approach, like adding some particular fertilizer, nutrient, or nutrient ratio of nitrogen phosphate to help the natural communities. Now, if there is an environment, for example, where for some reason there isn't a community of oil degrading bacteria that's so effective, then yeah, it makes sense that bioaugmentation could work. But you've got to think about, are the organisms that you're adding, which have come from a completely different environment, are the organisms you're adding to that environment going to survive? Are they going to tolerate that environment? So there's a lot of things that one needs to balance, and determine whether actually adding exogenous microbes could potentially work. I'm certainly not very much in favor of it.
Okay, thank you so much, sir. I think there are no more questions. So over to you, Kriti ma'am.
Thank you, Priyanka. We would be now officially concluding the session, and I would be formally thanking everyone who have joined us today. So first and foremost, it's my privilege to propose a vote of thanks on behalf of Department of Microbiology and the entire organizing team. And I would like to give a heartfelt thanks to our speaker, Dr. Tony Gutierrez, for such an enriching talk highlighting the huge and multifaceted influence of these invisible microbes on us in one or the other way. We are so grateful for your time and effort, sir. Thank you once again for this information and a great session. And we are privileged to have you today. And I express my profound gratitude to our principal, Dr. Pramila Kumar, for being appreciative and providing a great platform to organize this event. I would also like to thank our teacher in charge, Dr. Sashi Chawla, for giving me this opportunity. For her constant support and encouragement, her positive and uplifting attitude has been the driving force for us. I'm grateful to all our very motivated and dedicated students organizing team for their enormous cooperation and support in executing the event so efficiently and successfully. I'm appreciative of all our students for their active and enthusiastic participation. I also extend my gratitude to wonderful audience affiliated to different universities and academic institutes for their participation. Last but not the least, we have been very fortunate to be backed by a very motivated and dedicated technical support, Mr. Anuj Bhardwaj. So thank you, Anuj, for your support. Once again, I extend my gratitude to our speakers for the day, Dr. Tony Gutierrez, Dr. Christine Moissl-Echinger, our principal, Dr. Promila Kumar, teacher in charge, Dr. Sashi Chawla, faculty members, and everyone who has managed to join us today. Thank you very much for being with us. Please do connect with us tomorrow for a couple of beautiful sessions where we would be discussing the ways of managing waste, including the electronic waste. So, would you like to add before we wrap up? Dr. Tony, would you like to add before we wrap up?
Sorry, would I like to what? Sorry?
Would you like to add or say something before we
Yes.
wrap up?
Well, I just want to thank the organizers for inviting me. It's been a great pleasure to contribute a talk to this colloquium. It's a shame that it's so far away. I think it's probably my first colloquium talk that I've given to any university or college in India. So a great pleasure. And I hope we do it again. You do it again sometime.
Absolutely, sir. We're looking forward to it.
I'd be delighted to be invited, and I hope everyone keeps safe and we get through this pandemic, and maybe one day we can all meet.
Absolutely, sir. Wish the same, sir. Thank you so much. Sashi ma'am, you're on mute. Please unmute yourself.
Thank you, Dr. Tony, for being with us. It has been our privilege that you were here, and it was really an enlightening talk, and I hope that participants have whatever you have said. So thank you for being here with us, and we will definitely look forward to many more lectures from you also again. So thank you. And we are also sending you a potted plant. Yeah, Kriti, please tell him.
Yeah, so our gratitude token would be reaching you soon, sir. So please, I humbly request you to accept it.
Thank you. Thank you very much.
Okay, thank you, sir. Thank you for being with us. So we all can sign out now, everyone, and see you tomorrow, hopefully, for our day 2. Thank you very much, Dr. Tony. Sachi ma'am, you're on mute.
कल मिलते हैं then. We see each other tomorrow.
Yes. कल मिलते हैं at 3.